Close your eyes and imagine the smallest library in the world. Not the kind with wooden shelves and paper books, but something far more mysterious. Something that exists inside you right now, at this very moment. In every single cell of your body, there is a library. It is so small that millions of them could dance on the tip of a pin. Yet this tiny library holds more information than all the books ever written by humans. It contains the complete instructions for building you. This library is called DNA. And it exists not just in you, but in every living thing on Earth. Think of a hummingbird hovering outside your window, its wings beating faster than your heart. Inside each cell of that tiny bird lives the same kind of library. The books in its library contain different instructions than yours. They tell the hummingbird how to grow iridescent feathers, how to see colors you cannot imagine, how to navigate thousands of miles using only the stars. The oak tree in your yard has this library too. Its books explain how to grow roots deep into the earth, how to reach toward sunlight, how to live for hundreds of years, watching generations of creatures find shelter in its branches. Even the smallest ant carries this invisible library. Its books contain the wisdom of building underground cities, of working together in perfect harmony, of finding food and sharing it with thousands of sisters. All of these libraries are written in the same ancient language. A language with only 4 letters. Scientists call these letters A, T, G, and C. But you can think of them as nature's alphabet. With just these 4 letters, life writes every possible story. Your DNA library contains about 3 billion of these letters. If you could read them out loud, 1 letter per second, it would take you nearly a century to finish. The books would fill a stack taller than the Washington Monument. But here is the most beautiful part. You do not need to read these books consciously. Your cells read them for you, every moment of every day. Right now, as you rest quietly, millions of tiny librarians inside your body are checking these books. They are following the instructions to keep your heart beating, to help you breathe, to repair any damage and keep you healthy. These cellular librarians never sleep. They work in perfect silence, following recipes written before the first human ever walked the earth. Some instructions tell your cells how to make proteins. Others explain how to fight off illness. Still others contain the blueprints for growing and healing. For most of human history, we could only wonder about these invisible libraries. We knew that children looked like their parents. We understood that plants grew from seeds. But we could not read the ancient books that made it all possible. Then, about 70 years ago, scientists discovered how to peek inside these libraries. They learned to read the 4-letter language of life. They found that the books are not written on paper, but on long, twisted ribbons called chromosomes. Picture a spiral staircase made of letters, twisted and coiled like a spring. Each step of the staircase is made of 2 letters that fit together perfectly. A always pairs with T, G always pairs with C. This creates a ladder that can copy itself, passing information from parent to child across generations. The more scientists learned to read these books, the more amazed they became. They discovered that humans share many of the same chapters with other creatures. Nearly half of your library is identical to the library inside a banana. You and a chimpanzee share almost all the same books. Even tiny bacteria have some of the same basic instructions that keep you alive. This means that all life on Earth is connected through these ancient libraries. We are all reading from different versions of the same great book. The story that began billions of years ago, when the first simple cell learned to copy itself. But scientists did not stop at reading these libraries. They began to dream of something that seemed impossible. What if they could edit the books? What if they could help correct mistakes in the instructions? What if they could add new chapters to help creatures survive in a changing world? For many years, this remained just a dream. The books were too small, too complex, too delicate to change safely. It was like trying to edit a book written in letters smaller than atoms, using tools the size of mountains. Then something extraordinary happened. scientists discovered that nature had already invented the perfect editing tools. Deep in the ancient world of bacteria, they found molecular scissors that could cut and paste the letters of life with incredible precision. These tiny tools had been working for millions of years, helping bacteria defend themselves and adapt to new challenges. But now, scientists realized they could use these same tools to edit the libraries of other creatures. To help coral reefs survive warming oceans. To strengthen butterflies for their long migrations. To protect endangered species from disease. The name of these molecular scissors is CRISPR. And they represent the beginning of a new chapter in the ancient story of life on Earth. As you drift toward sleep tonight, your own invisible library continues its patient work. 3 billion letters, quietly maintaining the miracle that is you. Soon, we will discover how scientists learn to work alongside nature's own editors, opening new possibilities for all the wild creatures that share our world. In the deepest quiet of the night, when the world holds its breath, imagine the most delicate pair of scissors ever made. Not scissors of metal or plastic, but scissors so small they could dance between the letters in your DNA library. Scissors that work with the precision of starlight touching water. These scissors have a name that sounds like a whisper. CRISPR. Say it softly to yourself. CRISPR. It stands for something long and scientific, but you can think of it simply as nature's own editing tool. A gift that bacteria have been using for millions of years, and that scientists have only recently learned to borrow. Long before humans walked the earth, bacteria faced a problem. Tiny viruses would invade their cells, trying to rewrite their genetic libraries with harmful instructions. The bacteria needed a way to defend themselves. They needed to cut out the dangerous parts and repair the damage. So, over countless generations, bacteria developed the most elegant solution. They created molecular scissors that could recognize enemy DNA and cut it out with surgical precision. They also built a memory system, storing pieces of virus DNA like photographs in a police file. If the same virus ever returned, the bacteria would recognize it instantly and cut it out again. This ancient defense system became CRISPR. And for millions of years, it worked quietly in the microscopic world, protecting bacteria from harm. No human knew it existed. It was nature's secret, written in letters too small to see. Then, in laboratories around the world, scientists began studying these tiny bacterial warriors. They discovered something remarkable. The bacterial scissors could be taught to cut any DNA sequence they were shown. Not just virus DNA, but any genetic instructions at all. Imagine having a pair of scissors that could find one specific word in a library of 1 billion books. And not just find it, but cut it out and replace it with any word you chose. This is the miracle that scientists found hidden in bacteria. Two scientists, Jennifer Doudna and Emmanuel Charpentier, realized what this could mean. They saw that these molecular scissors could become tools for healing. Tools for helping creatures survive in a changing world. Tools for rewriting the stories of life itself, one careful letter at a time. Their discovery earned them the Nobel Prize. But more importantly, it opened a door to possibilities that had lived only in dreams. The scissors work like the most patient editors in the world. First, they are given a guide. This guide is like a bloodhound that can sniff out one specific sequence among billions of genetic letters. It searches through the twisted ribbons of DNA until it finds exactly the right spot. When the guide finds its target, it signals the scissors. The scissors move in with incredible gentleness. They make a clean cut, like separating two pages that have been glued together. The cell's own repair systems then spring into action, ready to fix the gap. But here is where the real magic happens. Scientists can provide new instructions to fill the gap. They can correct mistakes that cause disease. They can add helpful traits that make creatures stronger or more resilient. Or they can simply remove harmful sequences, letting the cell heal itself. The whole process happens in a space smaller than you can imagine. If your body were the size of the Earth, these molecular scissors would be smaller than a grain of sand. Yet they work with more precision than any human-made tool ever created. Scientists have learned to make these scissors even more precise. They have created versions that can make tiny changes, switching just one letter in the genetic alphabet. Other versions can turn genes on or off, like dimming lights instead of turning them off completely. Some can even add completely new instructions, expanding the library with helpful new chapters. But perhaps the most beautiful part of CRISPR is how gently it works. Unlike older genetic tools that were like using a sledgehammer to hang a picture, CRISPR works with the delicacy of a butterfly landing on a flower. It makes changes so small and precise that the cell barely notices the repair happening. In laboratories that stay quiet through the night, scientists are now using these bacterial scissors to help wild creatures in ways that were impossible just a few years ago. They are exploring how to help coral reefs resist warming oceans, how to strengthen butterflies for their incredible journeys across continents, how to protect endangered species from diseases that threaten their survival. Each possibility requires years of careful study. Scientists must understand not just how to make changes, but whether those changes will truly help the creatures they want to protect. They work with the patience of gardeners, knowing that the most important changes happen slowly, with great care and wisdom. The scissors themselves are not alive. They are tools, like a pencil or a paintbrush. But in the hands of thoughtful scientists, they become instruments of hope. Ways to help life adapt to challenges it has never faced before. As you rest tonight, these molecular scissors are working in laboratories around the world. Scientists are learning to read the genetic languages of creatures great and small. They are discovering which letters spell strength, which ones spell resilience, and which ones hold the keys to survival in tomorrow's world. The bacterial defenders that created these tools continue their ancient work, invisible and patient. They remind us that nature has always been the greatest inventor. And sometimes, the most powerful tools come not from human cleverness, but from learning to listen to the wisdom that life itself has discovered over millions of quiet years. Soon, we will visit the coral reefs where these scissors are offering new hope to underwater gardens threatened by warming seas. Imagine floating weightlessly in the clearest blue water you have ever seen. Sunlight filters down through the gentle waves above, creating dancing patterns of gold on the seafloor below. All around you, a magical city rises from the ocean depths. This is a coral reef. And it is one of the most beautiful rebellions in the natural world. Each coral looks like a tiny underwater flower, but it is actually an animal. A soft, gentle creature that builds its own house from minerals dissolved in seawater. Day by day, grain by grain, millions of these small architects create structures more magnificent than any human city. But corals hold a secret that makes them even more remarkable. Inside their translucent bodies live billions of microscopic partners called zooxanthellae. These tiny golden algae are like solar panels that capture sunlight and transform it into food. In return, the coral provides them with a safe home and the nutrients they need to grow. This partnership has flourished for millions of years. The algae paint the corals in brilliant colors. Electric blues, sunset oranges, deep purples, and soft greens that shimmer in the filtered sunlight. Together, they create underwater rainbows that stretch for miles. These coral cities become home to thousands of other creatures. Bright yellow tang fish dart between the branches like living flowers. Sea turtles glide gracefully overhead, their ancient eyes watching over the reef. Tiny cleaner fish set up stations where larger fish come to be groomed and healed. But today, these underwater gardens face a challenge they have never encountered before. The oceans are growing warmer. Not quickly, like the heat of summer, but slowly and steadily, degree by precious degree. When water becomes too warm, something heartbreaking happens. The coral becomes stressed, and the beautiful partnership begins to break down. The coral expels its algae partners, losing both its color and its main source of food. The vibrant reef becomes ghostly white, like a garden covered in snow. Scientists call this coral bleaching. And it is happening more often as our planet warms. In laboratories near the ocean's edge, marine biologists work through quiet nights, studying these underwater partnerships. They have discovered that some corals are naturally more resistant to heat than others. These hardy corals carry genetic instructions that help them stay cool under pressure. Using the molecular scissors we learned about, scientists are exploring whether they might share these cooling instructions with other corals. They work with the gentleness of underwater gardeners, studying which genetic letters spell resilience in the face of warming seas. The process begins with understanding exactly how heat tolerance works. Some corals have genes that help them produce protective proteins, like tiny umbrellas that shield their cells from damage. Others have instructions for building stronger partnerships with heat-resistant algae. In carefully controlled environments, scientists practice making small edits to coral DNA. They use CRISPR-like underwater writing tools, adding letters that might help corals stay calm when water's warm. Each change is tested slowly and thoroughly, the way a gardener might try a new fertilizer on just one plant before using it throughout the garden. But this work requires extraordinary patience. Corals grow slowly, like living stones. Changes that might show results in weeks with other creatures can take months or years to reveal themselves in coral. Scientists must wait and watch, measuring temperature tolerance degree by degree. The goal is not to create artificial corals, but to help existing reefs remember genetic skills they may have forgotten. Some coral populations once lived in naturally warmer waters. Their descendants might still carry the instructions for heat tolerance, written in genetic letters that just need to be awakened. Other scientists are exploring partnerships with the microscopic algae themselves. These golden partners also have genetic libraries that contain instructions for surviving heat. Perhaps the algae could be given better tools for staying healthy in warmer water, keeping their coral homes colorful and well-fed. The work happens in underwater nurseries where baby corals grow on special frames, like seedlings in a greenhouse. Scientists tend to these coral gardens with infinite care, monitoring temperature, light, and water chemistry. They watch for signs that their genetic gifts are helping the young corals grow stronger. Meanwhile, in the wild reefs, other scientists work to protect existing coral communities. They create shaded areas where corals can rest during the hottest parts of the day. They remove harmful algae that compete for space. They even relocate corals to slightly cooler waters, giving them time to recover and rebuild their strength. The most promising approach combines all these efforts. Scientists work to strengthen corals genetically while also protecting their homes and reducing the stresses they face. Like caring for a patient with both medicine and rest, the coral reefs need multiple types of healing. Some of the strongest hope comes from discovering that corals are natural survivors. They have weathered ice ages and warm periods throughout Earth's history. Their genetic libraries contain millions of years of wisdom about adapting to changing conditions. The challenge is time. Climate change is happening faster than corals can naturally adapt. But with gentle genetic assistance, these underwater cities might learn to rebuild themselves in tomorrow's warmer seas. As you drift towards sleep tonight, picture those coral reefs glowing softly in the deep blue darkness. Millions of tiny partnerships working together, building beauty from seawater and sunlight. Scientists tend to them like underwater gardeners, offering genetic gifts that might help these ancient cities survive and flourish. In the morning, we will follow the memory trails of elephants, where ancient wisdom written in DNA carries stories from the frozen past into an uncertain future. In the endless white silence of Siberian winter, something remarkable lies sleeping beneath the frozen ground. Deeper than tree roots reach, deeper than the summer sun can warm, ancient giants rest in ice that has not melted for thousands of years. These are the mammoths. Gentle cousins of today's elephants, who once walked through forests of ice and snow. Their massive bodies, covered in thick auburn fur, roamed across landscapes that stretched from horizon to horizon, painted in shades of white and gray. Picture one of these magnificent creatures in your mind. Imagine her standing against a backdrop of endless tundra, her breath forming clouds in the Arctic air. Her curved tusks catch the low winter sunlight. Her small, wise eyes reflect the patience of creatures who know how to survive when the world grows cold. The mammoths lived for thousands of years in harmony with the ice. Their DNA libraries contained detailed instructions for thriving in frozen worlds. They knew how to grow fur so thick that blizzards could not touch their skin. They understood how to find food beneath snow that covered the earth for months at a time. But about 4,000 years ago, the last mammoth walked her final steps. The ice began to retreat. Forests grew where tundra had stretched. The world grew warmer, and these cold-weather giants could not adapt quickly enough to survive. Or so we thought. In recent decades, scientists have made discoveries that read like stories from dreams. As the Siberian permafrost slowly melts, it reveals treasures beyond imagination. Perfectly preserved mammoth bodies, their fur still soft, their DNA still readable after millennia of sleep. These frozen time capsules contain genetic libraries written in ice. Scientists have learned to extract DNA from mammoth cells, throwing out instructions that have waited in silence for thousands of years. They have begun to read the ancient recipes for survival in cold worlds. Meanwhile, in warmer parts of our planet, elephants face challenges their species has never encountered before. Climate change brings unpredictable weather patterns. Droughts last longer than elephant memory can recall. Traditional migration routes disappear as human cities expand across ancient pathways. In laboratories that stay quiet through the night, geneticists work with the patience of archaeologists. They compare mammoth DNA with the genetic libraries of modern elephants. They discover something beautiful and surprising. The two species are like sisters, separated by time but not by blood. Elephants and mammoths share most of their genetic instructions. But mammoths possess special chapters in their DNA libraries. Chapters that explained how to grow thick, insulating fur. How to store extra fat for surviving long winters. How to digest tough, fibrous plants that grow in cold climates. Scientists wonder if these ancient survival skills might help modern elephants face tomorrow's challenges. Not by bringing mammoths back to life, but by sharing their genetic wisdom with their living cousins. The molecular scissors we have learned about offer a way to make this sharing possible. Researchers could potentially take the mammoth's instructions for growing thicker skin and give them to elephants who face increasingly harsh conditions. They could share the mammoth's ability to extract nutrition from difficult plants, helping elephants survive when their traditional food sources become scarce. But this work requires extraordinary care and wisdom. Scientists must ask deep questions about what it means to change a species, even to help it survive. They study elephant behavior, elephant families, elephant needs with the thoroughness of devoted students. Elephants are not just bodies that need survival traits. They are complex beings with rich emotional lives. They mourn their dead and celebrate new births. They remember family members across decades. They pass knowledge from grandmother to granddaughter through generations of gentle teaching. Any genetic changes must respect this complexity. Scientists work to understand not just which mammoth traits might help elephants survive, but which changes would honor the dignity and nature of elephant families. Some researchers focus on traits that would barely be noticeable to the elephants themselves. Slightly thicker skin that provides better protection from extreme heat and cold. Enhanced ability to digest cellulose, helping elephants extract more nutrition from the plants they already eat. Others explore whether mammoth DNA might help elephants resist diseases that threaten wild populations. Mammoths developed immunity to certain illnesses during their thousands of years in challenging environments. These genetic shields might protect modern elephants from new threats. The most ambitious scientists dream of using mammoth genetics to help elephants adapt to environments where they might otherwise struggle to survive. If climate change makes traditional elephant habitats too hot or too dry, perhaps mammoth traits could help elephants establish new homes in cooler regions. But every possibility is studied with the care of someone handling ancient treasure. Scientists test each idea in computer models first, then in laboratory cells, then in carefully controlled studies that take years to complete. They work with elephant experts, conservationists, and ethicists to ensure that any changes would truly benefit the elephants. The frozen mammoths continue to teach us even in their long sleep. Each new discovery reveals more about how creatures adapt to changing worlds. Scientists have found mammoth DNA in creatures as small as bacteria that lived in the mammoth's digestive system. Even these microscopic partners carried genetic instructions for surviving in ice. Some of the most interesting research happens in the spaces between species. Scientists study how mammoths and early elephants might have shared genetic material naturally, through rare interbreeding thousands of years ago. They look for evidence that evolution itself sometimes borrows survival strategies from cousins and ancestors. As this work continues, wild elephants live their lives with the same grace and wisdom they have shown for millions of years. In African savannas and Asian forests, elephant families continue their ancient migrations. Mothers teach daughters where to find water during dry seasons. Grandmothers remember paths through landscapes their great-grandmothers once walked. The genetic libraries of both elephants and mammoths hold memories of ice ages survived, droughts endured, and countless generations of careful adaptation. These memories, written in the four-letter language of DNA, whisper stories of resilience that span thousands of years. Tonight, as you rest quietly, mammoth DNA continues its patient sleep in Siberian ice. Elephant families gather around water holes under star-filled skies. And scientists work by lamplight, reading ancient genetic stories and wondering how the wisdom of the past might guide the survivors of tomorrow. Soon we will follow a different kind of ancient journey, as monarch butterflies carry their own genetic maps across continents that shift and change beneath their delicate wings. Close your eyes and imagine the most delicate traveler in the natural world. She weighs less than a paperclip, her wings thin as tissue paper painted in sunset orange and midnight black. Yet this tiny creature is preparing for a journey that would challenge the strongest birds. She is a monarch butterfly. And written in her genetic library are navigation instructions more precise than any human-made map. Every autumn, millions of monarchs begin one of nature's most remarkable migrations. From gardens and meadows across North America, they lift into the sky like living confetti. They ride air currents with wings that seem too fragile for such an epic journey. Their destination lies thousands of miles away. A small patch of mountain forest in central Mexico, where their great-great-grandparents once spent the winter months in gentle hibernation. No monarch butterfly has ever made this journey before. Yet somehow, each one knows exactly where to go. Picture her now, our tiny traveler, as she begins her flight. She rises above suburban neighborhoods where children once watched her emerge from her chrysalis. She soars over farmlands golden with harvest, over rivers that reflect the autumn sky, over cities that stretch toward distant horizons. She carries no GPS device, no paper map, no compass made of metal and glass. Instead, her navigation system is written in the very structure of her being. Her antennae sense magnetic fields that flow around the Earth like invisible rivers. Her eyes detect patterns of polarized sunlight that humans cannot see. Most remarkably, she possesses an internal clock so precise it would make Swiss watchmakers weep with envy. This clock tells her not just what time it is, but where the sun should be at each moment of her journey. As the sun moves across the sky, she adjusts her flight path with mathematical precision. But climate change is rewriting the maps that monarchs have followed for thousands of years. Weather patterns shift like restless tides. Storms arrive earlier or later than expected. The flowering plants that provide fuel for the journey bloom at different times, in different places. Some monarchs find themselves confused, their ancient navigation systems struggling to interpret a world that no longer matches their genetic expectations. They arrive at traditional feeding grounds to find flowers that have already faded, or seeds that have not yet bloomed. Scientists who study these remarkable navigators work with the gentleness of butterfly gardeners. They want to understand how the monarch's genetic compass works, and whether small adjustments might help these travelers adapt to changing conditions. In laboratories that stay quiet through long nights, researchers examine the molecular machinery of butterfly navigation. They discover that monarchs possess special cells in their brains, cells that contain tiny crystals of a protein called cryptochrome. These crystals respond to magnetic fields and help the butterfly sense which direction leads home. The butterflies also carry genetic instructions for timing their departure. Deep in their DNA libraries are chapters that explain how to read the changing length of days, how to sense the dropping temperature that signals winter's approach. These timing systems have worked perfectly for millions of years. But as climate patterns shift, some populations of monarchs benefit from slight adjustments to their timing. Scientists explore whether CRISPR might help butterflies fine-tune their internal clocks, helping them time their journeys to match new weather patterns. The work requires extraordinary delicacy. Butterfly genetics are as intricate as butterfly wings themselves. Each change must be tested carefully, like adjusting a watch that keeps time for an entire species. Some researchers focus on the monarch's ability to detect magnetic fields. They study whether small genetic changes might make the butterfly's magnetic sensors more sensitive, helping them navigate more accurately when weather patterns disrupt their traditional routes. Others explore the butterfly's response to changing plant cycles. As flowers bloom at different times due to shifting seasons, monarchs need to adjust their feeding and breeding schedules accordingly. Gentle genetic modifications might help them synchronize with these new rhythms of the natural world. But perhaps the most important research focuses on the monarch's remarkable memory systems. These butterflies can remember not just where they are going, but where their ancestors found the best nectar sources, the safest resting places, the most protected winter homes. Scientists are discovering that monarchs pass some navigation information from parent to child through genetic inheritance. Like ancient sailing instructions written in DNA, these hereditary maps guide butterflies along routes their grandparents once flew. As climate change alters landscapes and weather patterns, some of these inherited maps become outdated. Researchers wonder if they might help monarchs update their genetic GPS systems, incorporating new information about changed migration routes or shifted seasonal timing. All of this work happens with deep respect for the monarch's natural wisdom. These butterflies have been perfecting their navigation systems for far longer than humans have existed. Any genetic assistance must honor the elegance and precision of systems that evolution has refined over millions of years. In butterfly sanctuaries around the world, researchers raise monarchs with the care of devoted parents. They study how genetic changes affect flight patterns, navigation accuracy, and the butterfly's ability to find suitable winter homes. Each monarch is treated as precious cargo carrying the future of her species. As you rest tonight, imagine millions of monarch butterflies settling into their winter homes in mountain forests far to the south. Their wings fold like tiny prayer books, their bodies entering the quiet stillness of hibernation. Their genetic compasses rest, but do not sleep. In spring, their descendants will begin the journey northward, following maps written in DNA and adjusted, perhaps, by the gentle hands of scientists who understand that even the smallest creatures carry wisdom worth preserving. Soon, we will visit laboratories where other tiny creatures are learning to carry healing instead of harm, transforming from vectors of disease into messengers of hope. In the gentle twilight hour when day surrenders to night, a tiny hum fills the warm air. It is a sound that has followed humans since the beginning of time. The delicate wings of mosquitoes, beating faster than your heart, carrying these ancient creatures on their nightly journeys. For thousands of years, this sound has brought worry. Mosquitoes have carried diseases that have changed the course of human history. Malaria, dengue fever, yellow fever, and Zika have traveled the world on gossamer wings, causing suffering in communities from tropical rainforests to busy cities. But tonight, imagine a different story. Picture laboratories where scientists work with infinite patience, teaching these tiny creatures a new way of being in the world. The mosquito is not naturally cruel. She is simply following instructions written in her genetic library millions of years ago. When she lands on your skin with a touch lighter than a whisper, she is not seeking to harm. She needs protein to nourish the eggs developing inside her body. She is, in her own small way, a devoted mother preparing for the next generation. The problem is not the mosquito herself, but the passengers she sometimes carries. Microscopic parasites and viruses hitchhike in her bloodstream, traveling from person to person without the mosquito's knowledge or consent. She becomes an unwilling messenger, spreading illness when all she intended was to feed her young. Scientists have spent decades studying these intricate relationships. They have discovered something remarkable hidden in the mosquito's genetic code. Instructions that could transform these creatures from carriers of disease into guardians of health. In laboratories that hum with quiet dedication, researchers work with mosquitoes raised from eggs smaller than pinheads. They study these insects with the care of librarians examining ancient manuscripts. Every detail of mosquito biology is mapped and understood, from the compound eyes that detect movement, to the needle-like mouthparts that pierce skin so gently you barely feel the touch. Using the molecular scissors we have learned about, scientists explore ways to rewrite the mosquito's genetic story. They work to remove the chapters that allow harmful parasites to survive inside the mosquito's body. They add new paragraphs that help mosquitoes resist infection entirely. Some researchers focus on making mosquitoes inhospitable to malaria parasites. The malaria organism needs specific conditions inside the mosquito to complete its life cycle. By changing those conditions through careful genetic editing, scientists can break the chain of infection without harming the mosquito herself. Other teams explore even more ambitious possibilities. They wonder if mosquitoes might be taught to carry beneficial cargo instead of harmful parasites. Could these flying syringes deliver vaccines to remote communities where traditional medicine cannot reach? Could they become tiny healers, spreading immunity instead of illness? The work requires extraordinary precision. Mosquito genetics are as delicate as the creatures themselves. Each change must be tested carefully in controlled environments, ensuring that modified mosquitoes remain healthy and able to fulfill their natural roles in ecosystems. But the most promising research focuses on a phenomenon called gene drive. This is nature's own method for spreading genetic changes rapidly through a population. Scientists have discovered how to create genetic instructions that copy themselves, ensuring that beneficial traits pass to nearly all offspring instead of just half. Imagine releasing a small number of genetically modified mosquitoes into a wild population. Over just a few generations, the beneficial traits would spread like ripples across a pond, eventually reaching every mosquito in the region. Disease transmission could be reduced or eliminated entirely, without the need for pesticides or other interventions. The ethical implications of this work are as complex as the science itself. Scientists proceed with the caution of gardeners tending a delicate ecosystem. They study not just how to make these changes, but whether they should. They consult with communities that would be affected, ensuring that genetic modifications serve the needs of people who live with mosquito-borne diseases every day. Some researchers focus on self-limiting approaches. They create genetic modifications that fade away over time unless continuously renewed. This allows communities to try the technology, evaluate its benefits and risks, and choose whether to continue its use. Others work on precision targeting. Instead of changing all mosquitoes in an area, they focus on specific species known to carry particular diseases. Aedes aegypti mosquitoes spread Zika and dengue. Anopheles mosquitoes carry malaria. By targeting only disease-carrying species, researchers can reduce harm while preserving the beneficial roles that other mosquitoes play in ecosystems. In field stations around the world, scientists test their genetic modifications in large, screened enclosures that allow natural mosquito behavior while preventing escape into wild populations. They watch modified mosquitoes interact with plants, predators, and each other, studying how genetic changes affect the delicate balance of nature. The most encouraging results come from experiments with sterile male mosquitoes. Scientists use genetic engineering to create males whose offspring cannot survive to adulthood. When these modified males mate with wild females, mosquito populations decline naturally over time, reducing disease transmission without permanent genetic changes to wild populations. As this research continues, communities affected by mosquito-borne diseases participate as partners rather than subjects. Local knowledge guides scientific efforts. Traditional methods of mosquito control are combined with genetic approaches, creating comprehensive strategies tailored to specific environments and cultures. The goal is not to eliminate mosquitoes entirely. These tiny creatures serve important roles as pollinators for certain plants and as food for birds, bats, and other insects. The aim is more subtle and more compassionate. To help mosquitoes become the creatures they might have been in a world without the diseases they accidentally carry. As you rest tonight, imagine millions of mosquitoes beginning their evening flights. Some carry the ancient burden of disease, following genetic instructions written long before humans existed. But others, touched by science and guided by compassion, carry different cargo. They bring the possibility of healthier communities, fewer tears, and more children who can play safely in the warm twilight air. In the morning, we will visit meadows and gardens where other small creatures work as nature's essential partners, gathering nectar and pollen as they dance their ancient patterns between flowers. In a wildflower meadow kissed by morning sunlight, a small drama of survival unfolds with each gentle breeze. Thousands of wild bees move through the air like living jewels, their bodies dusted with golden pollen, their wings catching light as they dance from bloom to bloom. These are not the honeybees living in human-made hives, but their wild cousins. Solitary bees who make their homes in hollow stems and underground burrows. Bumblebees with fuzzy bodies that can fly in cooler weather than their smaller relatives. Carpenter bees who excavate perfect circles in dead wood, creating nurseries for their young. Each bee carries an invisible promise written in her genetic library. The promise to pollinate the flowers that feed the world. 1/3 of everything humans eat depends on these tiny workers. Apples and almonds, squash and strawberries, coffee beans and chocolate all exist because bees transfer pollen from flower to flower with the dedication of ancient gardeners. But these essential workers face challenges that their ancestors never knew. Pesticides drift like invisible fog across landscapes, confusing the navigation systems that guide bees home. Diseases spread more quickly as wild bee populations cluster around the few remaining patches of diverse flowers. Climate change shifts blooming seasons, creating gaps between when bees emerge from winter rest and when their favorite flowers open. Picture a bumblebee queen awakening from her winter sleep beneath a fallen log. She has slept alone through months of snow and ice, her body powered by fat reserves built up during the previous summer. Now, as spring warmth touches the earth, she must find flowers immediately to fuel her body for the enormous task ahead. She will spend the next week searching for the perfect place to start her colony. An abandoned mouse hole, perhaps, or a cavity beneath tree roots. There, she will lay her first eggs and tend them with the care of a devoted mother. She will gather pollen and nectar to feed her growing family, and teach her daughters the locations of the best flowers in the neighborhood. But what if the flowers she remembers are no longer there? What if the pesticides used on nearby farms make her forget the way home? What if new diseases attack her colony just as it begins to thrive? Scientists who study wild bees work with the patience of bee watchers themselves. They spend long hours in meadows and gardens, following individual bees to understand their needs. They map the flowers that different species prefer, the nesting sites they choose, the distances they travel to gather food. In laboratories where silence supports the most delicate work, researchers examine the genetic libraries of wild bees. They search for the chapters that explain how some bees resist diseases that devastate others. They study the instructions that help certain species tolerate cold weather or drought. They look for the genetic recipes that allow some bees to detoxify pesticides that poison their relatives. Using molecular scissors with the precision of master craftsmen, scientists explore whether genetic gifts from resilient bee populations might help struggling species survive modern challenges. They work with reverence for creatures whose work makes human civilization possible. Some research focuses on disease resistance. A fungus called nosema affects bee digestive systems, making it impossible for them to absorb nutrients from the pollen they collect. Scientists have discovered that some wild bee species carry genetic instructions for producing compounds that fight this infection. These natural medicines, written in DNA, might be shared with vulnerable bee populations. Other scientists study cold tolerance. As climate patterns shift, some bee species find themselves emerging from winter at the wrong times. Early warm spells trick them into activity before flowers are ready. Late spring freezes catch them unprepared. Genetic modifications that fine-tune temperature sensitivity might help bees synchronize their life cycles with changing seasons. The most promising research examines navigation systems. Wild bees possess internal compasses that rival any human technology. They use landmarks, polarized light patterns, and even the Earth's magnetic field to find their way home after foraging trips that can span miles. But pesticides can damage these navigation systems, leaving bees lost and unable to return to their nests. Scientists are learning to strengthen the genetic foundations of bee navigation. They study the molecular machinery that processes visual landmarks and magnetic information. Small genetic adjustments might make these systems more resilient to chemical interference. But perhaps the most important work focuses on genetic diversity itself. Wild bee populations need variety in their genetic libraries to adapt to new challenges. When habitats shrink and bee populations become isolated, this diversity disappears like colors fading from a painting. Researchers work to preserve and restore genetic diversity in wild bee communities. They use molecular techniques to understand which populations carry the most valuable survival traits. They develop strategies for reconnecting isolated bee communities, allowing beneficial genes to flow between populations like streams joining a river. All of this research proceeds with deep respect for the complexity of bee societies. Wild bees have evolved intricate relationships with specific plants, nesting sites, and seasonal patterns. Any genetic modifications must honor these ancient partnerships rather than disrupting them. Scientists test their approaches in contained environments where modified bees can be studied safely. They monitor not just survival rates, but behavior, reproduction, and the quality of pollination services. They ensure that genetic changes help bees fulfill their ecological roles more effectively, not just survive in isolation. The work happens in partnership with conservationists who protect wild bee habitats and farmers who provide the flowers and nesting sites that bees need. Genetic approaches complement rather than replace traditional conservation methods. As you rest tonight, wild bees settle into their hidden homes throughout the landscape. Some sleep in individual chambers they have excavated in sandy soil. Others cluster together in small colonies tucked away in tree cavities or abandoned rodent burrows. Their genetic libraries hold the wisdom of millions of years of pollination partnerships. Tomorrow, these essential workers will wake to continue their ancient dance between flowers, carrying life from bloom to bloom. Scientists work quietly in their laboratories, hoping to ensure that this dance continues for generations yet to come. In the distance, salmon are beginning their own ancient journey, carrying genetic memories upstream toward spawning grounds their ancestors knew for thousands of years. Listen to the sound of water moving over stones. Gentle, persistent, eternal. This is the voice of a mountain stream that has been singing the same song for thousands of years. But if you listen carefully, you might hear an echo of something missing. A splash that once punctuated the water's rhythm. The sound of salmon coming home. Picture a river in autumn, when maple leaves drift like golden coins on the current. Once, this waterway would have been alive with silver bodies leaping upstream against the flow. Chinook salmon, their backs dark as midnight, their bellies bright as moonlight. Coho salmon with hooked jaws and fierce determination. Sockeye salmon turning crimson red as they approached their ancestral spawning grounds. These remarkable fish spent their lives wandering the vast ocean, growing strong on a diet of krill and small fish. But when the time came to create the next generation, an ancient calling written in their genes brought them home. They would swim hundreds of miles against powerful currents, leaping over waterfalls, navigating by scent so faint that human noses could never detect them. Each salmon carried in her genetic library a map more detailed than any chart. The exact chemical signature of the stream where she was born. The precise temperature and mineral content of the water that first touched her scales. These memories, encoded in DNA, guided her through the ocean's immensity back to the exact gravel bed where her own mother once laid eggs. But in many rivers, the salmon have been absent for decades. Dams block their passage upstream. Pollution changes the chemical fingerprints they rely on for navigation. Climate change alters water temperatures and seasonal patterns. The ancient cycle of departure and return has been broken, leaving rivers that remember salmon but no longer welcome them home. In laboratories where hope meets science, researchers work as genetic archaeologists. They study museum specimens and tissue samples preserved from salmon runs that ended generations ago. From these silent witnesses, they extract DNA libraries that contain the navigation instructions of lost populations. These genetic maps tell stories of incredible specificity. Salmon from different tributaries of the same river system carry distinct genetic markers, like postal codes written in their cells. Even streams separated by just a few miles hosted salmon populations with unique genetic signatures, fine-tuned over thousands of years to local conditions. Scientists discover that salmon populations lost their genetic libraries gradually, not all at once. As dams were built and habitats degraded, some tributaries lost their salmon first. Others held on for decades longer. Each disappearance erased a unique chapter from the book of salmon genetics, knowledge that had accumulated over millennia. But the most remarkable discovery comes from studying salmon populations that survived in distant waters. These fish, descendants of the same ancestral stock, still carry genetic echoes of their extinct relatives. Hidden in their DNA are backup copies of navigation instructions, dormant but intact. Using the molecular scissors we have learned about, scientists explore whether these genetic memories might be awakened. They study the specific genes that encode olfactory recognition, the ability to detect and remember water chemistry. They map the genetic foundations of homing behavior, the mysterious force that draws salmon upstream toward home. The work requires patience measured in salmon generations. These fish take years to mature, and their complex life cycles cannot be rushed. Researchers raise salmon in carefully controlled hatcheries, studying how genetic modifications affect their ability to navigate, survive, and reproduce. Some experiments focus on enhancing the salmon's sensitivity to chemical cues. Modern rivers often carry pollutants that mask the natural scent salmon use for navigation. Genetic modifications that sharpen olfactory abilities might help salmon detect their home waters despite chemical interference. Other research explores temperature tolerance. Climate change is warming many salmon streams beyond the range these cold-water fish can survive. Scientists study genetic variants that allow some salmon populations to thrive in slightly warmer conditions. These heat-tolerance genes might be shared with struggling populations, helping them adapt to changing thermal environments. But perhaps the most ambitious work involves reconstructing lost genetic diversity. When salmon populations disappear from a watershed, they take with them thousands of years of local adaptation. Scientists work to understand which genetic traits were specific to particular river systems, and whether these can be restored through careful genetic archaeology. The process resembles detective work more than traditional science. Researchers examine tissue samples from salmon caught decades ago, before populations crashed. They compare these genetic profiles with surviving populations from similar environments. Slowly, they piece together the genetic recipes that made salmon successful in specific watersheds. Armed with this knowledge, scientists can begin to design restoration programs. They select breeding stock that carries the closest genetic matches to extinct populations. Using precision gene editing, they fine-tune traits like thermal tolerance, disease resistance, and homing ability to match historical conditions. But the salmon themselves provide the most important lessons. In the few places where runs have naturally recovered, scientists observe how quickly these fish readapt to their ancestral homes. Within just a few generations, salmon populations begin to show genetic changes that improve their success in local conditions. This remarkable adaptability suggests that salmon carry within their genes a vast library of potential traits, most dormant at any given time. Environmental pressures can activate different genetic programs, allowing populations to fine-tune themselves to local conditions with surprising speed. As restoration efforts continue, some rivers are beginning to hear familiar sounds again. The splash of salmon leaping waterfalls. The gentle disturbance of gravel as fish prepare spawning beds. The subtle but unmistakable signs that ancient cycles are beginning to renew themselves. These returning salmon carry more than their own genetic libraries. They bring marine nutrients far inland, feeding forests and wildlife that have been deprived of ocean-derived nitrogen for generations. Their decomposing bodies enrich stream ecosystems, supporting everything from insects to bears. The work continues in laboratories and field stations around the world. Scientists perfect techniques for genetic restoration while conservationists remove barriers and restore habitats. Each small success teaches lessons that can be applied to other watersheds, other species, other broken cycles waiting for renewal. As you rest tonight, imagine streams flowing quietly through darkened forests, their waters carrying the chemical signatures that salmon once knew by heart. In some distant tributary, perhaps the first genetically assisted salmon are already learning to recognize the scent of home. Soon, we will travel to wilderness areas where other ancient partnerships are being renewed, as predator and prey rediscover their roles in the patient dance that keeps ecosystems in balance. In the blue hour before dawn, when shadows still hold the forest in their gentle embrace, a lone howl rises through the mountain air. It is a sound that has echoed across wilderness landscapes for thousands of years. Deep, mournful, beautiful. The voice of the wolf, calling to family scattered across miles of territory. This ancient song carries more than longing. It speaks of a creature whose very presence shapes the world around it. The wolf is what scientists call a keystone species. Like the central stone that holds an arch in place, wolves hold entire ecosystems in delicate balance through their patient work as nature's gardeners. Picture a valley before wolves return to it. Deer and elk graze without fear, staying in the same meadows for weeks at a time. They eat young trees down to stumps, preventing forests from growing. Rivers run straight and narrow because there are no tree roots to slow their flow. The landscape becomes simplified, less diverse, less alive. Now imagine wolves walking quietly back into this valley. Their presence alone changes everything, even before they hunt. Deer become alert, moving frequently, never staying in one place too long. Young trees begin to grow in the meadows where deer once lingered. Streams start to meander as growing forests create natural barriers and root systems. This is called a trophic cascade. The effects of predators ripple through the entire food web like gentle waves. Wolves hunt elk, which allows willows to grow, which creates habitat for songbirds, which eat insects that might otherwise damage other plants. Each connection strengthens the whole. But wolves face challenges that test the ancient wisdom written in their genes. Habitat fragmentation divides wolf territories into islands too small to support healthy packs. Genetic bottlenecks occur when wolf populations become isolated, reducing the diversity needed for long-term survival. Climate change shifts the ranges of their prey species, requiring wolves to adapt their hunting strategies and territory patterns. In research stations where scientists work with the dedication of wildlife guardians, geneticists study the complex inheritance systems that make wolves successful. They discover that wolf genetics are far more intricate than simple traits like coat color or body size. The genetic foundations of pack behavior, hunting coordination, and territorial navigation involve hundreds of genes working together in precise harmony. Some of the most important research focuses on genetic diversity within wolf populations. Healthy wolf genetics require what scientists call heterozygosity. This means having different versions of important genes, providing flexibility to adapt to changing conditions. When wolf populations become too small or isolated, this diversity disappears like colors fading from a sunset. Scientists use molecular techniques to measure genetic health in wolf populations across different regions. They can identify populations at risk of genetic bottlenecks before problems become visible in the wolves themselves. This early warning system allows conservationists to take action while there is still time to preserve genetic diversity. One approach involves facilitating gene flow between isolated populations. Scientists study historical migration patterns to understand how wolves naturally move between territories. They work with wildlife corridors and land management agencies to restore connections that allow genetic material to flow between populations like streams joining a river. In cases where natural gene flow cannot be restored, researchers explore more direct interventions. They might relocate individual wolves from genetically diverse populations to areas where genetic diversity has declined. These immigrant wolves bring new genetic variants, refreshing the local gene pool and reducing the risks associated with inbreeding. The most cutting-edge research explores how CRISPR technology might help preserve crucial genetic diversity. Scientists can identify specific genetic variants that are disappearing from small wolf populations. Using precision gene editing, they might restore these variants to breeding programs, ensuring that important genetic tools for survival do not vanish forever. But this work requires extraordinary care and wisdom. Wolves are not just collections of useful genes. They are complex social beings with intricate behavioral patterns that depend on genetic, environmental, and cultural factors working together. Any genetic interventions must preserve the essential nature of what makes wolves successful as pack hunters and ecosystem engineers. Researchers study how genetic traits relate to behaviors that define wolf society. The ability to form strong pack bonds. The coordination required for successful group hunts. The communication systems that allow wolves to maintain territories spanning hundreds of square miles. These complex behaviors emerge from interactions between many genes, making genetic interventions challenging and delicate. Some of the most promising work focuses on disease resistance. Wolves face threats from diseases like mange, distemper, and parvovirus that can devastate small populations. Scientists have identified genetic variants that provide natural immunity to some of these diseases. Ensuring that these protective genes remain present in wolf populations could prevent future outbreaks from causing extinctions. Climate adaptation represents another frontier for wolf genetics research. As climate patterns shift, the prey species that wolves depend on may change their distributions and behaviors. Wolves with genetic flexibility in hunting strategies and territorial preferences may be better equipped to adapt to these changes. The research happens in partnership with indigenous communities whose traditional knowledge spans thousands of years of living alongside wolves. These partnerships combine genetic science with cultural wisdom, ensuring that conservation efforts respect both biological needs and traditional relationships with the land. Field researchers spend years observing individual wolves, documenting how genetic differences translate into real-world survival and reproduction. They use non-invasive techniques to collect genetic samples from scat and shed fur, building detailed family trees that reveal how genetic traits move through wild populations. As this work continues, some wolf populations show encouraging signs of recovery. Genetic diversity increases as isolated populations reconnect. Pack behaviors strengthen as genetic bottlenecks are resolved. The subtle but crucial effects of healthy predator populations begin to ripple through ecosystems. The ultimate goal is not to engineer better wolves, but to ensure that these remarkable creatures retain the genetic tools they need to continue their ancient role as ecosystem gardeners. Their patient work maintains the complexity and resilience that keep wilderness landscapes healthy and whole. Tonight, wolf packs settle into hidden dens scattered across remote territories, their genetic libraries holding millions of years of predator wisdom. Tomorrow, their descendants will continue the delicate work of maintaining balance in places where wildness still remembers its own rhythms. In the quiet spaces between predator and prey, another ancient relationship waits to be renewed, as forests themselves prepare to grow stronger roots in tomorrow's uncertain climate. Stand quietly among ancient trees and feel the profound silence that only old forests know. Above you, branches interweave like the fingers of old friends, creating a living ceiling that filters sunlight into gentle green light. Beneath your feet, roots spread through dark soil in networks more complex than any human city, sharing resources and information in languages we are only beginning to understand. These trees have stood here longer than human memory reaches. They have weathered countless storms, survived droughts that lasted years, and provided shelter for thousands of generations of woodland creatures. Their genetic libraries contain chapters written across centuries, stories of resilience that span climate cycles humans never witnessed. But today's forests face challenges that no tree in history has encountered. The air carries higher levels of carbon dioxide than any plant has known for millions of years. Temperatures rise beyond the ranges that shaped these ancient genetic programs. New diseases spread faster through interconnected forests than ever before. Even rainfall patterns shift, creating floods and droughts in places where water once fell predictably. In research stations tucked quietly among the trees they study, forest geneticists work with the patience of the forests themselves. They spend decades watching individual trees, measuring how different genetic variants respond to changing conditions. They climb into canopies to collect seeds from the strongest survivors, studying the genetic recipes that allow some trees to thrive while others struggle. These scientists have discovered something remarkable about forest genetics. Trees possess genetic flexibility that exceeds almost any other form of life. A single tree might live for hundreds of years, experiencing climate conditions that change dramatically over its lifetime. To survive such long lives, trees carry genetic programs for responding to almost any environmental challenge. Some genetic variants help trees tolerate drought by growing deeper roots or closing their leaf pores more efficiently. Others provide resistance to fungal diseases that attack weakened trees during stressful periods. Still others allow trees to take advantage of higher carbon dioxide levels, growing faster and stronger than their ancestors could ever imagine. Using the molecular scissors we have learned about, scientists explore whether they might help trees access these beneficial traits more quickly than natural selection would allow. They work with the reverence of gardeners tending to living cathedrals, understanding that changes made today will echo through centuries to come. Some research focuses on helping trees adapt to higher temperatures. In carefully controlled tree nurseries, scientists study genetic variants that allow certain populations to thrive in warmer conditions. These heat-tolerance genes might be shared with tree populations currently struggling as their traditional climates shift northward or to higher elevations. Other scientists explore drought resistance. Trees that have evolved in naturally dry regions often carry genetic instructions for surviving with less water. These drought survival genes might help trees in regions where rainfall is becoming less reliable due to changing weather patterns. The most ambitious research examines carbon dioxide uptake. As atmospheric carbon levels rise, some trees show remarkable ability to take advantage of this abundance, growing faster and stronger than ever before. Scientists study the genetic foundations of this enhanced growth, wondering if these traits might help forests become even more effective at removing carbon from the atmosphere. Disease resistance represents another crucial frontier. New fungal infections spread rapidly through forests stressed by climate change. Trees weakened by heat and drought become vulnerable to diseases their healthy ancestors could resist easily. Scientists search for genetic variants that provide natural immunity to these emerging threats. But forest genetic research requires extraordinary patience. Trees grow slowly, and their responses to environmental changes unfold over years or decades. Scientists must design experiments that span human careers, passing research projects from one generation of scientists to the next like sacred trusts. The work happens in forest laboratories that exist both indoors and outdoors. Controlled environment chambers allow scientists to test how genetic variants respond to different temperature and moisture conditions. Meanwhile, field plots scattered throughout natural forests provide long-term data on how different genetic lines perform in real-world conditions. Some of the most promising research focuses on root systems. Tree roots form partnerships with fungal networks that help forests share resources and information. Scientists study genetic variants that enhance these partnerships, potentially helping trees access nutrients and water more efficiently while building stronger connections within forest communities. Seed banks preserve genetic diversity from forests around the world, storing genetic variants in careful climate-controlled conditions. These living libraries ensure that valuable genetic traits are not lost even if wild populations face temporary setbacks. Scientists can draw upon this stored diversity to help forests adapt to future challenges. The research combines cutting-edge molecular techniques with traditional forestry wisdom. Indigenous knowledge about forest management, passed down through generations, guides scientific efforts to understand how trees naturally respond to environmental stresses. This partnership between traditional and modern approaches creates more comprehensive strategies for forest conservation. Field researchers work in forests from tropical rainforests to boreal woodlands, studying how different tree species might be helped to adapt to local climate challenges. They collect samples from the strongest, healthiest trees, analyzing genetic patterns that correlate with survival and growth under stress. Perhaps most importantly, scientists study not just individual trees, but whole forest ecosystems. They explore how genetic changes in dominant tree species might affect the countless other organisms that depend on forests for habitat. Any genetic modifications must support the complex web of relationships that make forests living communities rather than simple collections of trees. As this research progresses, some forests are beginning to show signs of natural adaptation to changing conditions. Trees in areas experiencing the greatest climate stress sometimes evolve resistance to heat or drought within just a few generations. These rapid adaptations provide hope that forests possess the genetic flexibility needed to survive in a changing world. The ultimate goal is not to replace natural forests with genetically modified ones, but to help existing forests access their own genetic potential for adaptation. Like careful gardeners providing support for plants during difficult seasons, scientists work to ensure that forests retain the genetic tools they need to continue their ancient work as planetary guardians. Tonight, forests around the world settle into the quiet rhythms of darkness. Their roots continue their underground conversations. Their branches shelter countless sleeping creatures. Their genetic libraries hold millions of years of wisdom about survival, growth, and the patient work of creating oxygen from sunlight and soil. As these forest guardians continue their ancient work, questions arise about the wisdom and responsibility that guide all genetic research, challenges that require not just scientific knowledge, but careful consideration of what it means to hold such power over the living world. In the quiet moments before important decisions, wise people often seek places of stillness. They walk in old forests where ancient trees have witnessed countless seasons of change. They sit beside rivers that have flowed through landscapes for thousands of years. They listen to the wind, waiting for clarity to emerge from the deep well of contemplation. Today, scientists who work with the molecular scissors of genetic editing face decisions that require this same kind of deep listening. They hold in their hands the power to change the very essence of living creatures. With each careful cut and precise edit, they shape the future of species that have evolved over millions of years. This power brings with it questions that have no simple answers. Questions that echo through research laboratories and conservation centers around the world. Questions that require not just scientific knowledge, but wisdom, compassion, and humility. When is it right to change the genetic story of a wild creature? When does helping become interfering? How do we know whether our changes will bring healing or harm to the complex web of life that surrounds us? These questions become more urgent as the challenges facing wildlife grow more severe. Climate change moves faster than most species can naturally adapt. Human activities have created threats that no creature in evolutionary history has encountered before. Pollution, habitat loss, and emerging diseases push many species toward extinction. In this context, genetic editing appears like a life preserver thrown to drowning swimmers. Scientists can potentially help coral reefs survive warming oceans, protect elephants from new diseases, guide butterflies through shifting migration routes. The molecular scissors offer hope for species that might otherwise face extinction. But with this power comes profound responsibility. Every genetic change creates ripple effects that spread through ecosystems in ways we cannot fully predict. A modification that helps one species might inadvertently harm another. Changes intended to solve immediate problems might create unexpected challenges for future generations. Scientists approach these dilemmas with the careful consideration of doctors taking an oath to first do no harm. They spend years studying each potential application, testing ideas in controlled environments before considering any release into wild populations. They consult with ethicists, conservationists, indigenous communities, and local people who live alongside the species being considered for modification. Some ethical frameworks focus on the concept of consent. Wild creatures cannot give permission for genetic modifications the way human patients can consent to medical treatments. This creates a special obligation for scientists to act only in ways that clearly benefit the animals themselves, not just human interests. Other ethical approaches emphasize precaution. They suggest that when the consequences of genetic modification are uncertain, it is better to proceed slowly and carefully rather than risk causing irreversible harm. This perspective values the wisdom of natural systems that have evolved over vast periods of time. Still, other ethical frameworks focus on justice and equity. They ask who benefits from genetic modifications and who bears the risks. Communities that live closest to wildlife often have the most to gain from successful conservation efforts, but they may also face the greatest consequences if genetic interventions go wrong. The most thoughtful scientists incorporate multiple ethical perspectives into their decision-making processes. They consider not just whether they can make genetic changes, but whether they should. They ask not only what is scientifically possible, but what is morally right. Some research focuses on modifications that closely mimic natural evolutionary processes. Scientists identify genetic variants that already exist within wild populations and help beneficial traits spread more quickly than natural selection alone might accomplish. This approach works with evolution rather than against it, accelerating natural adaptations rather than creating entirely artificial ones. Other research emphasizes reversibility. Scientists design genetic changes that can be undone if unexpected problems arise. Like experimental treatments that can be discontinued if they cause harmful side effects, these modifications include genetic switches that allow future scientists to reverse changes if necessary. The concept of genetic rescue provides another ethical framework. When species face imminent extinction due to genetic bottlenecks or environmental pressures, genetic modifications might be justified as emergency interventions. Just as doctors might use experimental treatments to save patients facing otherwise certain death, conservationists might use genetic editing to prevent extinctions. But perhaps the most important ethical principle involves listening deeply to the natural world itself. Scientists spend time observing wild animals in their natural habitats, learning about their needs, behaviors, and relationships. They study ecosystem dynamics to understand how genetic changes might affect the complex web of interactions that sustain life. Indigenous communities offer crucial perspectives on these questions. Their traditional knowledge systems, developed over thousands of years of living closely with wildlife, provide insights that complement scientific understanding. Many indigenous cultures view genetic modification through the lens of reciprocity and respect, asking whether proposed changes honor the intrinsic value of wild creatures. Religious and spiritual traditions also contribute wisdom to these discussions. Many faith traditions emphasize stewardship responsibilities, the obligation to care for the natural world as sacred trust. Others focus on humility, recognizing the limits of human understanding when it comes to the complexity of living systems. As these conversations continue, scientists develop increasingly sophisticated approaches to genetic research that honor both scientific possibilities and ethical responsibilities. They create advisory committees that include diverse perspectives. They engage with communities that would be affected by genetic modifications. They design research programs that can adapt and respond to new ethical insights as they emerge. The goal is not to resolve all ethical questions before proceeding with research, but to ensure that ethical considerations remain central to every decision. Scientists work to develop genetic technologies that reflect humanity's best impulses toward compassion, wisdom, and respect for the natural world. The questions themselves evolve as scientific capabilities advance. Today's ethical dilemmas about helping coral reefs adapt to warming oceans may seem simple compared to tomorrow's questions about more complex genetic interventions. But the commitment to thoughtful, ethical decision-making remains constant. As you rest tonight, picture scientists in laboratories around the world pausing in their work to consider these deeper questions. They set down their molecular tools and step outside to look at stars that have witnessed the evolution of every species they study. In the morning, we will explore how all these considerations come together in the quiet space where science meets wisdom, where human capability encounters the sacred responsibility of sharing this beautiful, complex planet with all its living inhabitants. In the gentle darkness before dawn, when the world rests between night and morning, dreams take their most vivid forms. This is the liminal hour when possibilities shimmer like dewdrops on spider webs, when the future feels both fragile and full of promise. It is in this quiet space that we come to understand the deepest truths about the genetic stories we have explored together. Throughout our journey, we have witnessed the remarkable power of molecular scissors to rewrite the ancient texts of life. We have seen coral reefs that might learn to survive in warming oceans. Elephants that could carry the cold weather wisdom of their mammoth cousins. Butterflies guided by enhanced genetic compasses through shifting migration routes. We have imagined mosquitoes transformed from carriers of disease into messengers of health. Bees strengthened against the challenges of changing seasons. Salmon returning to rivers that remember their genetic songs. Wolves maintaining the genetic diversity that allows them to serve as nature's gardeners. Each of these possibilities exists today not as reality, but as careful research unfolding in laboratories where scientists work with the patience of monks illuminating manuscripts. The technologies are real. The molecular tools function with precision that would have seemed magical to previous generations. The genetic maps are being drawn with increasing accuracy and detail. But as we reach the end of our exploration, we discover something unexpected. The most important genetic editing may not happen in laboratories at all. The changes that matter most may not require molecular scissors or microscopic precision. Instead, they may unfold through choices made by ordinary people who decide to see the world differently. Consider the genetic future of a coral reef. Yes, scientists may eventually help corals develop greater heat tolerance through careful genetic modifications. But the reef's ultimate survival depends more on human decisions about carbon emissions, ocean pollution, and coastal development. The most powerful genetic intervention might be choosing to protect the waters where corals live. Think about the monarch butterflies carrying their genetic maps across continents. Research may improve their navigation systems and climate adaptations. But their greatest genetic gift comes from people who plant native flowers along migration routes, who reduce pesticide use in their gardens, who create corridors of habitat that allow genetic diversity to flow naturally between populations. The salmon returning to rivers that remember their names may benefit from genetic restoration techniques. But their genetic libraries can only be expressed in streams that flow freely, in watersheds protected from pollution, in ecosystems where the ancient cycles of departure and return are honored and preserved. Even the molecular scissors themselves reflect this deeper truth. CRISPR technology emerged from studying how bacteria naturally defend themselves against viral infections. The most sophisticated genetic tool humans have ever developed came not from human engineering, but from learning to listen carefully to the wisdom already written in living systems. This suggests a different way of thinking about genetic editing and wildlife conservation. Instead of seeing ourselves as editors rewriting the book of life, we might imagine ourselves as librarians helping to preserve and protect the vast genetic libraries that already exist. Our role becomes one of stewardship rather than authorship. The most profound genetic modifications happen when we choose to leave space for wild creatures to be themselves. When we protect large enough habitats for natural genetic diversity to flourish. When we reduce the human pressures that force animals to adapt faster than their genetic systems can manage. When we create corridors that allow genes to flow naturally between populations. Sometimes the best genetic intervention is no intervention at all. It is the patient choice to step back and allow evolutionary processes to unfold at their own pace. It is the wisdom to recognize that genetic systems refined over millions of years often know better than we do how to respond to environmental challenges. But there are also times when careful genetic assistance represents the highest form of compassion. When species face extinction due to human-caused problems that natural selection cannot solve quickly enough. When genetic bottlenecks threaten to erase millions of years of evolutionary wisdom. When emerging diseases or climate changes outpace the ability of natural systems to adapt. In these moments, genetic editing becomes an act of humility rather than hubris. Scientists work not to improve upon nature, but to help natural systems access their own genetic potential. They serve as midwives for evolutionary processes, assisting adaptations that might otherwise arrive too late. The choices we make about genetic technology reflect deeper choices about our relationship with the natural world. Do we see ourselves as separate from nature, using our tools to manage and control wild systems? Or do we understand ourselves as part of the living web, using our unique capabilities to support the health and resilience of the whole? The answer may lie in remembering that we too carry genetic libraries written over millions of years. We are not outside the story of evolution, but characters within it. Our ability to develop molecular scissors and read genetic codes represents not a departure from nature, but nature's own experiment in consciousness and compassion. Perhaps the most important genetic editing happens in our own minds and hearts. When we choose to see wild creatures as relatives rather than resources. When we recognize that their genetic stories are connected to our own. When we understand that protecting their genetic diversity ultimately protects the genetic heritage we all share. As dawn approaches and our journey together draws to a close, imagine the genetic libraries of countless species continuing their quiet work around the world. In every cell of every creature, ancient wisdom persists and new possibilities emerge. the four-letter alphabet of life continues writing stories of adaptation, survival, and hope. The molecular scissors rest in their laboratories, tools of enormous power held in trust for the future. Scientists continue their patient research, guided by both wonder and wisdom. And in the spaces between human intervention and natural process, new chapters in the great book of life wait to be written. Tonight, as you drift toward sleep, your own genetic library continues its faithful work, maintaining the miracle that is you. And somewhere in the vast conversation between all living things, the deepest dreams of genetic editing unfold not in laboratories, but in the simple, profound choice to share this beautiful world with kindness, respect, and hope.